Hello and welcome back to another A-Level biology video with Starfish Revision. This one is all about DNA. By the end of this video, we'll have covered the structure of DNA and RNA nucleotides, the structure of DNA itself, how DNA replicates, and the Michelson and Stahl experiment into semiconservative replication. DNA stands for deoxyribonucleic acid. It's found in the nuclei of our cells and it holds all the genetic information that's needed to make a full organism. Our DNA exists in long pieces called chromosomes. There are 46 of these chromosomes in each human cell. DNA is a polymer, which means it must be made out of monomers. The monomers of DNA are called nucleotides. Let's look at the structure of a DNA nucleotide now. We have a pentose sugar called deoxyribose in the middle. Pentose means the sugar has 5 carbons. On one end of the sugar is a phosphate group, and on the other end is a base. A base is a nitrogen-containing group of atoms. There are 4 different bases: adenine, cytosine, guanine, and thymine, abbreviated to A, C, G, and T. RNA stands for ribonucleic acid. RNA comes in a variety of different forms, and we'll look at that further when we do protein synthesis. But for now, let's look at the structure of an RNA nucleotide. RNA also has a pentose sugar in the middle, but it's called ribose. There is a phosphate group on one side, just like in DNA, and on the other side is the base. RNA has the bases adenine, cytosine, and guanine, but instead of thymine, it has uracil. Here are the 2 nucleotides side by side so you can compare them. You'll need to be able to sketch them both in an exam. Now we'll look at how nucleotides join together in DNA. DNA has a double helix structure, which is basically a twisted ladder. The sides of the ladder are the sugar phosphate backbones. Each backbone is created when nucleotides join together in a condensation reaction. The bond that's formed between them is called a phosphodiester bond, and it forms between the sugar of one nucleotide and the phosphate group of the next. The rungs of the ladder are created when each base of one side pairs by hydrogen bonding with a base on the other side. Adenine always pairs with thymine, and cytosine always pairs with guanine. This is called complementary base pairing. There are 2 hydrogen bonds between adenine and thymine, and 3 hydrogen bonds between cytosine and guanine. If we look carefully, we can see that the 2 strands are running in opposite directions, or on the right-hand strand, it looks like the nucleotides are upside down. We say that the strands are anti-parallel. If we look back at the structure of a single DNA nucleotide, we can see that there are 5 carbon atoms making up the pentose sugar. The phosphate group is attached to carbon number 5. The little apostrophe after the number tells us to read this as 5'. So this is the 5' end of the nucleotide. When nucleotides join together, the bond forms between the third carbon and the phosphate group of the nucleotide below it. So we call this the 3' end of the nucleotide. And when many nucleotides are strung together, there will be a 3' and a 5' end to the chain. Because the strands are anti-parallel, the 3' and 5' ends will be on opposite sides of the molecule. The structure of DNA was discovered in 1953 by Watson and Crick, helped with the X-ray crystallography images by Rosalind Franklin. There are some links in the description if you're interested in the story behind the discovery of the structure of DNA. Now, a very important feature of DNA as a molecule is that it has the ability to replicate itself. Not many other molecules are able to do this. DNA needs to replicate itself every time the cell divides, so that the two daughter cells end up with the same genetic information as the parent cell. Let's see how DNA does this. First, we unzip the DNA. An enzyme called DNA helicase moves down the DNA molecule, breaking the hydrogen bonds between the bases as it goes. This exposes the bases on either side. Next, free DNA nucleotides, which are always floating around the nucleus, pair up with the exposed bases by hydrogen bonding. Complementary base pairing occurs, so A always pairs with T and C always pairs with G. Finally, another enzyme called DNA polymerase joins the new nucleotides together by phosphodiester bonds, creating new sugar phosphate backbones. So we create two new strands of DNA, each with one strand from the original DNA molecule and one new strand. This is therefore known as semiconservative replication. Remember the 3' and 5' ends? Well, we need to remember that DNA polymerase is only complementary to the 3' end of the newly forming strand. Therefore, it moves in the 5' to 3' direction down the newly forming strand. And so DNA polymerase moves in opposite directions. One DNA polymerase enzyme will be moving downwards while the other one is moving upwards. When the structure of DNA was first discovered, scientists predicted that it could replicate itself by semiconservative replication, but they didn't have any solid evidence for this until Michelson and Stahl performed this experiment. Michelson and Stahl grew 2 colonies of bacteria. One colony was grown in nutrient broth, which only contained a light isotope of nitrogen, that's nitrogen 14. And the other was grown in a nutrient broth that only contained heavy nitrogen, that's nitrogen 15. As the bacteria grew and divided, it used the nitrogen in the nutrient broth to make the bases for its new nucleotides, which would then be incorporated into their DNA as the DNA replicated. Michelson and Stahl then extracted the DNA from both colonies and spun them in a centrifuge. The DNA from bacteria grown in light nitrogen settled out further up the tube than the DNA from bacteria grown in heavy nitrogen. Next, they took some bacteria that only contained heavy nitrogen and they transferred it to a broth that contained only light nitrogen, and they allowed the DNA to replicate. They then extracted the DNA and centrifuged it like before. Now, if conservative replication was taking place, you would expect to see two separate bands of DNA because the original DNA would only contain the heavy nitrogen and the new DNA would only contain the light nitrogen. However, this did not happen. Instead, the DNA settled out somewhere in the middle of the tube, showing that the new DNA molecules contained half heavy nitrogen and half light nitrogen, supporting the theory of semiconservative replication. So that's it for DNA. I hope you found this helpful, and if you did, please feel free to like and subscribe, and I'll see you in the next video. Thanks for watching.